Today, we are exploring the dark matter of your DNA.
Yeah, which is just a wild concept to wrap your head around.
It really is. I mean, for decades, if you looked at a map of the human genome, the greatest minds in science would point to just like 1 or 2% of it.
Right, the parts that actually code for proteins.
Exactly, and they declare that tiny fraction to be the blueprint of human life. But the other 98%, it was literally dismissed as junk DNA.
Just evolutionary baggage.
Right. But a massive new study just proved that supposed junk is actually hiding these invisible managers that keep us alive, and hidden tripwires that might actually just cure cancer. So let's get into it.
It is genuinely staggering to think about how long we assumed that 98% was useless. I mean, the prevailing thought was that it was basically just transcriptional noise.
Like the genome just humming to itself.
Exactly, yeah, just background static.
But we know now that assumption couldn't have been more wrong. So our deep dive today relies on a really dense, groundbreaking 2026 paper.
Right, published in the journal Cell Genomics by Liang and this huge team of researchers.
Yeah, and their mission, which is our mission today, is to basically map out these hidden managers living inside that dark matter.
Because understanding them changes basically everything we know about how human life develops from a single cell.
Totally. And the scale of what they've done here is really what sets it apart.
For sure. I mean, we've known for a bit that the genome is pervasively transcribed, meaning the cell is constantly making RNA copies of those non-coding regions.
But this study goes further.
Way further. They systematically hunt down exactly which of those mysterious transcripts are fundamentally essential for a cell to even survive.
Okay, let's unpack this.
Yeah.
Because we are talking about a very specific type of molecule here called lncRNAs.
Right.
What exactly is happening on a biological level with these things? Like, I really want to break down every mechanism here.
Okay, so lncRNA stands for long non-coding RNA. To really grasp what they are, you have to think about the central dogma of biology.
Which is DNA makes RNA and RNA makes proteins.
Exactly. Proteins are the physical workers. They build the cell, they move things, they do the actual labor. Now, lncRNAs are transcribed from your DNA, just like normal messenger RNA.
Right.
The cell goes to all the trouble of unzipping the DNA, making a copy, editing that copy, capping the ends so it doesn't degrade.
So it's like a movie studio going through all the expense of writing a script, hiring actors, filming the movie, adding all these special effects, and then
they just lock the film in a vault.
Right. It never gets projected on a screen.
That is a brilliant way to frame it, actually. The transcript never goes to the ribosome to be translated into a protein. It just, stays as a raw RNA molecule.
And because they don't make proteins, they are notoriously difficult to study, right?
Oh, incredibly. They are low in abundance. Their sequences change rapidly over evolutionary time. For a very long time, scientists honestly couldn't easily distinguish them from random cellular garbage.
Which makes the scope of this source document so impressive. I mean, they didn't just look at 1 or 2 of these. They systematically analyzed roughly 500 different lncRNAs.
Yeah, about 5,496 to be exact.
Wow. And they didn't just test them in one environment either. They tested them across 5 radically different human cell lines.
Right, from HEP1, HEK293FT, K562, MDA, MB231, and THP1. So basically everything from a leukemia cell type to a breast cancer cell line.
Just to ensure their findings weren't some fluke in one specific tissue?
Exactly. Essentially, they went in and systematically deleted these lncRNAs one by one to see what would happen when these invisible managers go missing.
It's like the ultimate biological stress test.
You break a component and see if the engine still runs.
But hold on, if these molecules don't make proteins, they're basically invisible to a lot of our normal testing methods. How did Liang's team actually find them without, accidentally breaking the rest of the cell in the process?
That is the big question.
Right. Because haven't we been using CRISPR to edit genes for over a decade? Why is this specific hunt considered such a huge technological breakthrough?
So the distinction lies in the specific tool they deployed. Previous studies absolutely used CRISPR, but they primarily used CRISPR-Cas9, or a variation called CRISPR interference.
No problem what that is.
The fundamental limitation of Cas9 is that it targets DNA. When you use Cas9 to shut down an lncRNA, you are physically altering the master DNA at that specific location on the chromosome.
Oh, I see. And when you mess with the master DNA, you get collateral damage.
Inevitably. I mean, the human genome is incredibly dense. By physically mutating the DNA to turn off the lncRNA, you might accidentally shut down a completely unrelated vital protein-coding gene sitting right next door.
Right. Or disrupt a hidden regulatory element.
Exactly. You introduce massive confounders. If the cell dies, you have no idea if it died because it lost the lncRNA or because your DNA edit accidentally killed its neighbor.
Let me try to visualize this. If the human genome is this massive ancient recipe book, using Cas9 to get rid of a bad recipe is like literally ripping the entire page out of the book.
Yes.
You get rid of the recipe you wanted, gone, sure. But you might accidentally tear the vital recipe printed on the back of that same page. You've damaged the book itself.
That's a perfect analogy. And to avoid ripping the page, this team had to completely abandon Cas9 and use something else entirely, which is CRISPR-Cas13.
Cas13.
Cas13 is a different beast because it targets RNA, not DNA.
So instead of ripping the page out of the master recipe book, Cas13 is more like an interceptor.
Exactly, like an RNA sniper.
Right. It leaves the original ancient book completely intact on the pedestal. It just waits for the cell to make a temporary photocopy of the recipe, the RNA transcript. And while that photocopy is being carried down the hallway to the kitchen, Cas13 intercepts and shreds it.
That is the exact mechanism. It provides ultimate precision. Because the DNA remains untouched, the researchers know with absolute certainty that if the cell gets sick, it's strictly because that specific RNA transcript is missing.
Wow. No collateral damage to the neighbors.
None. And speaking of that extreme precision, there is this fascinating metascience detail hidden in the methods section of this paper.
Oh yeah, the correction thing.
Yes. This 2026 study actually exists to essentially correct and refine a paper these very same authors published back in 2025.
It really highlights how rigorous the scientific method has to be when you're dealing with millions of molecules.
Absolutely. In their earlier work, they realized that some of their guide RNAs, the molecular homing beacons they programmed to tell the Cas13 interceptor what to shred, were occasionally sticking to similar-looking off-target RNAs.
So they weren't quite precise enough.
Right. So for this updated study, they went back to the drawing board and ruthlessly filtered their targeting library. They removed any homing beacon that had even up to 2 mismatches with other known transcripts in the cell.
They completely recalibrated the targeting system to ensure absolute zero collateral damage.
Exactly.
Okay, let's look at the actual numbers this hyper-precise hunt yielded, because they're pretty wild. Out of the roughly 5,500 lncRNAs they targeted, the interceptor technique revealed exactly 788 that are, quote, essential.
Meaning if you shred them, the cell literally stops growing and dies.
The breakdown of those 788 essential molecules is where a really striking pattern emerges, right?
Yeah, a massive 82% of them, so 648 out of the 788, were essential in only one specific cell type out of the 5 they tested.
Wow. So over 80% are hyper-specialized.
Very specialized. Another 16%, about 125 of them, were partially shared, meaning they were vital in maybe 2 or 3 cell lines.
And the universally shared one?
Only a tiny fraction. About 2%, which is just 15 specific lncRNAs, were shared across 4 or more cell lines, things like MELAT21 and MIR17HG, which are heavily studied.
Okay, let's do a quick 5-minute recap of where we are, just to consolidate all this.
Sounds good.
Bullet 1. lncRNAs are non-coding managers that don't make proteins. Bullet 2. The researchers use Cas13, an RNA sniper, to shred them without touching the master DNA. And bullet 3. Out of thousands, they found 788 that are absolutely critical for a cell to survive.
Spot on.
So to you, the listener, think about this for a second. If you were a cell, would you rely on a manager that only works in one specific tissue or a universal boss? Keep that in mind as we look at where these lncRNAs actually live, because the vast majority of them only manage one specific type of tissue. Which begs a huge question, right? Who exactly are they managing?
That's the million-dollar question.
The most obvious assumption would be that they just boss around their immediate neighbors.
Right, and that has been a long-standing, very comfortable assumption in genomic science. The proximity myth.
Because they don't make their own proteins to send across the cell, people just figured they acted locally.
Exactly. The assumption was that their entire job was to regulate the transcription of whatever protein-coding gene happened to be sitting directly adjacent to them on the DNA strand.
But this paper tests that local supervisor myth by looking at thousands of neighboring pairs.
What's fascinating here is they systematically analyzed 4,013 pairs, looking at a specific lncRNA and its closest protein-coding neighbor. And the data just completely shatters the proximity myth.
It's highly asymmetric, isn't it?
Very. For instance, in the HAP1 cell line, they found 534 cases where either the lncRNA was essential or its protein-coding neighbor was essential. But they found only 28 pairs where both of them were essential to the cell.
That overlap is basically a rounding error. And the source highlights a specific case that makes this really tangible.
Oh, yes. XLOC047247.
Right. It's a highly essential lncRNA manager. If you send the case 13 interceptor to shred it, the cell dies immediately. But if you look at its immediate neighbor on the DNA strand, which is a protein-coding gene called CCM2, and you knock that out, the cell doesn't care at all. It survives just fine.
And they saw the exact reverse, too. The lncRNA XLOC025611 is completely useless to cell survival, but its neighbor PSIC4 is absolutely essential.
It proves that they aren't functioning as a unified team just because they share a zip code on the genome.
Exactly.
But hold on, though. The genome isn't a flat, straight line. It's three-dimensional. It's folded up inside the nucleus like a microscopic bowl of spaghetti.
Right, exactly.
So even if they aren't next to each other linearly on the strand, couldn't the DNA loop around so that they're reaching across the room and physically touching a vital gene in 3D space?
That's a great point, and the researchers anticipated that exact counter-argument. To solve it, they used a highly advanced technique called H3K27UI-CHE.
Okay, rather than getting bogged down in the acronym, how does that actually work? How do you map the spaghetti ball?
You essentially freeze the cell. You use chemical cross-linkers to act like molecular glue, freezing the DNA in its folded state and gluing together any pieces of the chromatin that are physically touching each other in 3D space.
Oh, wow. So you freeze it in place.
Right. Then you extract that glued DNA and sequence it to map out the contacts.
That is wild.
And even when they built this 3D spatial map, they found that only between 4 and 27% of these essential lncRNAs directly contact essential protein-coding genes.
So the vast majority do not.
Exactly. They aren't local bystanders. They have their own global agendas in the cell.
Okay, so connecting the dots here. If they aren't local bystanders and they aren't just reaching across the 3D space, it means these essential lncRNAs are highly independent operators.
Yes.
They evolved to do their own vital jobs entirely independent of who they're sitting next to. Which brings us to the most dramatic part of the research, in my opinion. The emergency brakes.
This is where it gets really intense.
Right. If these independent managers are keeping the cell alive, what exactly happens inside the cell the very moment our Cas13 interceptor shreds them?
To watch that happen in real time requires a level of technological granularity that almost sounds like science fiction.
Oh, totally. Because they couldn't just look at a whole dish of millions of cells and measure the average response. They needed to see the exact cascading failure inside individual microscopic cells.
Here's where it gets really interesting. They used an approach called carpool-seq.
Carpool-seq, what does that stand for?
Cas13 RNA perturb-seq. What makes this so mind-bending is the scale. They're introducing the Cas13 interceptor into a massive population of cells to target 27 specific essential lncRNAs.
Okay.
But then they sequence the entire transcriptome, meaning they read every single piece of RNA being produced in single individual cells simultaneously.
Carpool-seq allows you to trace the exact biological consequence of the knockout cell by isolated cell.
Right. And the data reveals a brutal immediate reaction. When the interceptor hits the target lncRNA, the cells experience severely impaired cell cycle progression, and they drive aggressively toward apoptosis.
Apoptosis being programmed cell death.
Yes. The cell actively dismantles itself.
The analogy that comes to mind isn't a sniper anymore, it's more like a massive factory floor.
I like that, yeah.
Your cell cycle is this series of incredibly complex assembly lines, right? Copying DNA, organizing resources, preparing the cell to physically divide. The lncRNAs are the floor managers coordinating the unseen logistics.
Exactly.
If you suddenly remove those managers, the assembly line doesn't just slow down, it aggressively jams up. Cellular products crash into each other. And when a biological factory jams, it doesn't just sit there idle, it initiates a self-destruct sequence so it doesn't mutate into a rogue cancerous mass.
And the mechanism driving that self-destruct is one of the most famous systems in biology, the p53 pathway.
Right.
The researchers saw this pathway light up across the board. The p53 pathway is the cell's ultimate emergency brake for DNA repair and apoptosis.
The guardian of the genome. But how exactly does shredding an RNA manager pull that emergency brake? I mean, on a molecular level.
The single-cell data shows us it happens in two distinct ways. In some cases, losing the lncRNA caused the cell to directly upregulate the TP53 transcript itself.
So it literally just manufactured more of the emergency brake to force the stop.
Exactly. But the other mechanism is even more fascinating. It involves downregulating a gene called MDM2.
And what role does MDM2 play in the factory?
MDM2 usually acts as a primary suppressor of P53. If P53 is the emergency brake handle, MDM2 is the heavy plastic safety cover you have to break to pull it.
All that makes so much sense. It keeps the brake from being pulled accidentally during normal operations.
Exactly. The lncRNA normally helps keep that safety cover intact. So by removing the lncRNA, you downregulate MDM2. You essentially take the safety off the emergency brake, allowing the P53 pathway to activate and execute the stalled cell.
Okay, let's step back into our second small recap to look at the whole board for a second.
Let's do it.
Bullet 1. We found out these lncRNAs operate completely independently of their neighbors. Bullet 2. Removing them with our Case 13 tool jams the cell cycle. And bullet 3, that jam triggers the P53 emergency brake, killing the cell.
Perfect summary.
So to the listener, think about this. Your cells have hundreds of invisible tripwires. But why would the body evolve such an intricate network of non-coding managers in the first place? To answer that, we have to pull all the way back from the microscopic level.
Way back.
We have to look at the massive macroscopic extremes of human biology, embryonic birth, and terminal cancer.
You really do have to look at those extremes because these essential lncRNAs aren't just doing mundane maintenance in adult cells. They are fundamentally tied to massive, coordinated, explosive cellular growth.
So they looked at development.
Right. To prove this, the research team took their new list of 788 essential lncRNAs and cross-referenced it against a massive developmental atlas.
This atlas is incredible, by the way. It's built from 182 human tissue samples, spanning a timeline from just 4 weeks post-conception all the way to advanced old age.
It's essentially a high-resolution time-lapse of human development, and the correlation is undeniable. These essential lncRNAs are highly expressed very early in embryonic development.
In specific areas.
Oh yeah, you see massive spikes of these transcripts in the developing brain, the early heart, the forming liver and kidney. But as the tissue matures and we age, the expression of these lncRNAs significantly decreases.
So they are the specific managers called in for early rapid construction.
Exactly. And the paper notes they correlate incredibly tightly with known cellular proliferation markers, specifically pCNA and MKI67.
Okay. Those markers are technically the biochemical exhaust fumes of explosive cell growth.
Yep. Where you see those markers, you see these lncRNAs calling the shots.
But how does an RNA spike actually translate to building a liver, though?
Well, it goes back to that factory floor assembly line. Building an organ from scratch requires millions of cells to divide in perfect synchrony, migrating to the right places, avoiding premature apoptosis.
That's a lot of coordination.
Huge amounts. These lncRNAs are the logistical software running in the background, ensuring the assembly lines run at maximum capacity without triggering the P53 emergency brakes. Once the organ is built, the factory slows down, and you don't need those heavy-duty logistics managers anymore. Their expression drops.
Which logically brings us to the dark side of cellular growth. Because what is cancer, biologically speaking, if not embryonic level cell growth, happening at the completely wrong time, in the wrong place?
If we connect this to the bigger picture, it is the exact same biological machinery, just hijacked. The researchers knew they had to look at tumors, so they performed a massive computational alignment.
Using what data?
They took RNA sequencing data from over 8,800 primary tumors across 29 different cancer types, utilizing the Cancer Genome Atlas, or TCGA.
8,800 tumors. I mean, the sheer computing power needed to cross-reference thousands of lncRNAs against the entire transcriptomes of nearly 9,000 tumors is staggering.
Yeah.
And what did they find hiding in all that cancer data?
They found that these essential lncRNAs are heavily differentially expressed in tumors compared to adjacent, healthy, normal tissue.
So they are turned back on.
Exactly. In many highly aggressive cancers, the tumors have essentially reached into the genome, found these dormant developmental managers from when we were embryos, and turned them back on to drive their own rogue growth.
There is a specific case study in the paper that really anchors this concept, involving a universally shared lncRNA called PVT1, right?
Yes, PBT1 is a prime example of this hijacking. It's one of those rare, universally shared, essential lncRNAs. When the researchers looked at the clinical patient data, they found that the expression levels of PBT1 directly correlate with survival rates in several very serious malignancies.
Which one specifically?
Specifically, lower-grade glioma in the brain, uveal melanoma in the eye, and kidney renal clear cell carcinoma.
Meaning the higher the levels of this non-coding RNA in the tumor, the worse the prognosis.
Drastically worse. If a patient's tumor had highly elevated levels of PDT1, their survival rate dropped significantly. The tumor was fundamentally relying on that resurrected manager to suppress the p53 emergency bruk and survive its own unchecked replication.
Wait, if the tumor is deeply addicted to this specific RNA, and we now have the technology to intercept and destroy specific RNAs, couldn't we theoretically just build a drug that acts exactly like the Case13 interceptor we used in the lab?
You have just articulated the massive clinical payoff of this entire field of research. Historically, we've looked at RNA primarily as a biomarker.
Like a clue left behind at a crime scene that helps us diagnose the cancer.
Exactly. But this paper proves that these lncRNAs are actual mechanical vulnerabilities. They are therapeutic targets.
So what does this all mean for patients? How do we hit them without using CRISPR on a live human being?
We can design therapies called antisense oligonucleotides, or ASOs.
Okay, let's break that down. What is an ASO?
An ASO is a synthetic, custom-built molecule. You design it in the lab to perfectly match the sequence of the target RNA. You infuse it into the patient, it enters the cells, and it binds like Velcro to that very specific RNA transcript.
And then what?
Once it binds, it signals the cell's own natural enzymes to just shred the transcript.
It's like a synthetic assassin.
Yeah.
So if we can map out exactly which lncRNA a specific patient's tumor is addicted to, we deliver an ASO tailored to that exact sequence.
Yep.
It enters the tumor, knocks out the hijack manager, jams the cancer's assembly line, and the tumor's on p53 emergency brake finally activates. The cancer cell is forced to undergo apoptosis and self-destruct, all without us ever touching or altering the patient's healthy master DNA.
It is a profound paradigm shift in how we approach targeted therapy. It proves that the complexity of human life and the diseases that threaten it rely just as much on the RNA that isn't translated into protein as the RNA that is.
That is just wild.
For decades, we have been staring at a fraction of the blueprint. We are only just beginning to decipher the real, hidden operating manual of the human cell.
That is absolutely incredible. I mean, we started this deep dive talking about junk DNA. And look at the conceptual journey this 2026 paper just put us on.
It's a huge leap.
We went from viewing the vast majority of our genome as useless transcriptional noise to utilizing hyper-precise RNA interceptors to identify 788 independent, highly specific molecular managers.
Managers that literally hold the keys to a cell's survival.
Right, that drive our earliest embryonic development and that dictate whether a tumor lives or dies. It completely rewrites how we view the dark matter of our own biology.
It really does.
Which leaves us with one final thought for you, the listener, to mull over today. For decades, the greatest minds in biology largely ignored these regions of our DNA simply because they didn't make proteins. They didn't fit the expected central dogma mold. But if we just found 788 essential life or death managers hiding in the junk, simply because we finally used the right tool to look for them, what else is hiding in the remaining 98% of your genome, just waiting for someone to invent the right tool to discover it?
Something to think about.
Thanks for joining us on this deep dive. We'll see you next time.